to share screen. Can everybody hear me?
I can do.
That works. This whole K-storm thing, I'm learning new things every day between the children being homeschooled, between this. I will be technically up to date even if I don't want to. But I'm going to share my screen now. Okay. And there we go.
Looking good. Yeah, we got that. Thank you.
It's looking good. All right. First of all,
minutes.
I want to say thank you so much for inviting me to come and talk to you guys today about these extraordinary animals that I've had the good fortune of studying for the past 20, 25 years now. And what I want to do today is I want to talk about the tools, the innovation, the energy that we are going to need to adopt to try and find a ways to predict future pandemics and to prepare for them. And I'm going to argue with you and discuss with you why studying bats will help us do this. And really, I'll have this facetious title: Are bats the cause, the cure, and the future of all pandemics, or just this one? And just for everybody in the audience, that is a vampire bat. There are 3 species of vampire bats in the world, and this is one of them. But what I want to do before I start into what we can do to predict and prevent pandemics is tell you a little bit about these fantastic mammals. So why do we want to study bats? Why should anybody study bats? If you think about it, why would you study one group of the 9 million eukaryotes on our planet? Why is it important to study bats? Well, they're fascinating creatures. So you can see here from this picture, 1 in 5 of every living mammal on this planet is actually a bat, and they've huge diversity. There's about 1,400 species and more. So there's lots and lots of bats, and they're found everywhere throughout the globe. And potentially one of the reasons for their brilliant success is that they fly. And so bats are the only true flying mammal. Nothing else flies that's a mammal. They fall with style, but they do not fly. Bats fly. And that means that they can move around. So there's something to consider. They can move around much more easily than terrestrial non-flying mammals. They are nocturnal for the most part. There's about 2-3 diurnal species, and they're able to use these unique form of sensory perception. So they use hearing to orient in complete darkness. And with this, they come with these magnificent, weird facial structures, great big ears and nose leaves, like you can see here on this horseshoe bat. And so the majority of bats use echolocation, but you have one family of bats, the pteropods, these big, large fruit bats that you'll find in Australia, for example, that do not use laryngeal echolocation, but rather have large eyes and a keen sense of smell, but they're also nocturnal and feed on fruit. So if you want to understand the evolution and the genomic basis of sensory perception, bats are a cool order to study. But more importantly, they are key to our ecosystems functioning. In the tropics, bats are major pollinators and also seed dispersers. The majority of bats in temperate parts of the world are insectivorous. And what that means is that they will feed on the pests of crops and the pests of us, and they maintain balance in our ecosystems. And it's been estimated that if we were to lose bats, one species of bats alone from the United States, it would cost the US taxpayer about $3 billion every year in taxes to pay for insecticides to do the job that bats do. So we need bats. We can't wipe them all out, even if they are potentially scary and carry lots of viruses. Also, a lot of work that I'm not going to talk about today, but I think is directly related to bats' unique immune response, is bats do not age. They do not get cancer. They are the longest lived mammal given their body size. And studying how bats have evolved mechanisms to slow down the aging process can usually help us. But in relation to our pandemics, bats seem to have a very unique immune system. And so bats have now come to the fore in light of SARS-CoV-2 and COVID-19 that has really devastated the whole world. But for a long time, people who were researching bats realized that bats seem to be able to carry and tolerate a lot of viruses that are typically deadly to humans. It's been argued that they may be reservoirs for Ebola. They are known reservoirs for SARS, for MERS-like coronaviruses, potentially for Marburg. The jury's out on whether or not bats can actually survive rabies or not. So some research say yes, some research say no. But without a doubt, they have a very unique immune response that allows them to tolerate and live with these pathogens. And the question is, what is the relationship with COVID-19, with this disease that we are now experiencing globally? And I want to give you an example of how studying bats in terms of their relationship with SARS-CoV-2 that can help us predict and prevent future pandemics. And really studying bats from an ecological viewpoint with the most modern genomic tools, with most modern surveillance tools, with the newest methods to allow us to analyze the data we have, will allow us address this grand challenge of emerging infectious diseases. And this is just showing you, it's the past 2 weeks of this global accumulation of COVID-19 and how it's increasing throughout the world. And we need to try and deal with this. And we also need to make sure that this doesn't happen again. But what is our relationship with bats and with COVID-19? Why bats? Now, there's been loads and loads of propaganda in the news. Even when I was in my local supermarket when this whole thing happened, I had people crying, saying they weren't going into the park because they saw a bat there 4 years ago. But what is the relationship, the real relationship with bats and with SARS-CoV-2? Well, the highest diversity of coronaviruses have been found within bats. And there's lots of work being done by the Predictive consortium and so forth, that you do find a high diversity of coronaviruses when you sample bats. And this is a paper that was published in 2014. And this is a diversity that you see of different coronaviruses here. And what you see that in red are all of the coronaviruses that were found within bats. And you can see they seem to have this huge diversity of coronaviruses, which suggests that they can survive and live with coronaviruses. And indeed, when we were scrambling, trying to find where this new SARS-CoV-2 came from, there was lots of work done, and it was identified that in this clade here, the SARS-CoV-2, here are the human SARS-CoV, and the closest ancestor to the human clade is the bat coronavirus, or ATG13. And you can see there's the Rhinolaevis malayanus, there's another one there. What you find when you do phylogenetic analysis of the whole genome of the SARS-like coronaviruses, that the closest relative to the human SARS-CoV-2 is found within bats, but it's not identical. So 96% of the SARS-CoV-2 is found here with this. It is similar to the rat TG13. So they're not identical, but they're very similar. And indeed, it's been estimated using really state-of-the-art phylogenetic analysis. And again, I would recommend anybody should read this paper in how do you analyze recombining coronaviruses using phylogenetic methods. It's quite excellent. But that SARS-CoV-2 diverged from its bat ancestor, or ATG13, 40 to 70 years ago. So yes, they're very closely related. And yes, in the different wildlife that we have sequenced and that we have sampled and surveyed for SARS-like coronaviruses, the closest one is in bats, but we're missing 40 to 70 years of evolutionary history. So there's something going on here that we need to understand. So work was done by EcoHealth Alliance, by Kevin Olival and a lot of their groups, and Kendra Phelps. And it was like trying to work out, where did SARS-CoV-2 come from? And there's work that's done. This is this excellent surveillance that has to happen, where you'll take droppings and you look at this 440 base pair fragment of a particular gene. And when this was done, what was found that here's SARS-CoV-2, here you've got the pangolins, and here you have different species of these horseshoe bats, Asian horseshoe bats. And you find that this is where it came from. And so by looking and surveying and sampling across bats, we can find the origin of these zoonotic pathogens. So it does look like it's coming from bats from what we've sampled so far. So potentially bats are a reservoir of the sarbecoviruses. So what this means that we need to survey bat populations to predict and understand potential future pandemics and work out where the next zoonotics might be coming from. But how do you do this? Well, first of all, you're going to have to work with the bat researchers. You have to work with the ecologists who know the behavior, the biology, and have studied these populations for years. And this is just an example of a fantastic study that's been going on for 60 years in the UK on Rhinolophus firmequinum, greater horseshoe bat, run by Roger Ransom and Garrett Jones. But it takes a huge amount of dedication. And you need to have these neat longitudinal marker capture field studies that allow us understand the dynamics of the viruses within the population. You need, though, to link this with new molecular methods. And these are the innovations I think we need to think of in this session. So old-fashioned, hardcore field biology that's been going on for a long time, simple marker capture studies, being able to model our understanding of how the viruses are changing the dynamics within the populations, but we need new molecular methods. Because think about it, we're not going to be able to lethally sample all of these bats. You have populations sometimes in the millions in certain regions, but you will be able to take droppings and potentially non-lethal swabs. And the problem with this is that these are trace amount of samples. And even when people have enriched for sequencing viruses with shotgun sequencing, they'll find that only 1% of what is returned is actually virus. There's a lot of this viral dark matter. And so the reality is that we need to develop the methodology to be able to really sequence deeply viruses from trace samples if we're going to do this. And bats leave droppings. Anybody ever had bats in their attic knows that this is the case. So we can do it. We also need to look to our biodiversity collections. So people around the world have been collecting samples in museums with some of these wonderful collections in Canada, in the ROM, whereby you have access to samples taken a long time ago. So again, if you can develop and optimize ways to detect and sequence viruses from these samples, we will have a timeline of what was the viral landscape like at the time. So we need to use these. But we also need to develop better molecular methods to be able to survey from droppings. So most of the time when we're screening for the likes of SARS-like coronaviruses, typically people will look for this region of the viral genome. But the reality is all these different pieces of the genome are very, very important. And what we need to do is we need to be able to sequence the entire viral genome, and we need to be able to assemble it from trace samples. And these are the methodologies that we need to move forward on, how we can sequence and assemble genomes from really, really trace amounts of droppings, for example, or saliva samples. And this will help us move forward. Also, we need to have better phylogenetic methods. We need to uncover the evolutionary history of the host and of the pathogen. And again, the idea is that if you can find the closest relative, for example, of Rhinolophus affinis, potentially you could see what has the virus changed like within these individuals. This will allow us, the phylogenetic trees will allow us to predict which species we could look at. We also need to have exquisite, brilliant methods of analyzing the viruses themselves. So we need to get up to date with the appropriate molecular evolutionary models to make evolutionary trees to better predict. But again, why bats? So why are they important? So this is the cause, but what about the cure? So bat immune system has come under great scrutiny. And again, there seems to be unique adaptations that bats have. So the question is, are bats more likely to produce a zoonotic event? And some argue yes, and some argue no. But the idea is that potentially bats' immune system has predominantly the antiviral mechanism switched on all of the time. And they couple that with an equally aggressive anti-inflammatory response. And this may act as a selective pressure on the viruses, causing them to change, and change maybe allows them hop between hosts. This is a theory. Research that we've done, when we go and we look and we've isolated back macrophages and challenged them with immune agonists. And what you'll find is that bats typically will mount an extremely aggressive antiviral response, but they dampen that with an even more aggressive anti-inflammatory response. And you can look at these different cytokines. This is stimulated bat macrophages. And what we found in Ireland, that when they're trying to predict the outcome of patients that turn up when they have COVID-19, is that if they have an immune cytokine ratio more similar to bats than typically humans, it looks like they're not going to go on a ventilator. So this is how you can study bats who've naturally evolved mechanisms to tolerate these viruses. And so how can you use bats then as an alternative model system to allow us to uncover the molecular pathways that underlie their extraordinary disease tolerance? And this is the work that I've been involved in for a long time. But if you want to do this, if you really want to use bats to understand how we can handle viruses, you need to have exquisite genomes. And to do this, I set up Bat1K's consortium with my co-director, Sonia Vernies. And what we want to do is we want to sequence the genome of every single living bat species to chromosome level error-free assemblies. To do this will allow us to uncover the genomic basis of their unique traits, their extraordinary adaptations, and ultimately drive global conservation and awareness. There's a group of people here. Here's the other co-founding director, Sonia Vernies, and these are members of the executive, David Ray from Texas Tech, Michael Hiller from Max Planck in Germany, Jean Myers from Max Planck, and Lillian Davalos from Stony Brook in the USA. And we all came together and thought, you know what, this is a great idea, let's do this. And we spent about 4 or 5 years trying to optimize how we do this. How do you get the samples? How do you move them around? How do you sequence them? We worked together with the brilliant genome technologists, with phylogenomicists, and we developed the tools and the methods to be able to sequence error-free, as close as we can, genome assemblies of bats. And this paper, this research, our pilot study got published in Nature just in July. And the question was, well, all right, if you sequence these genomes, can you find any evidence of bats' unique immunity? And it was extraordinary, because you can. And so what we found, if you just look at this here, was that, for example, this is the apobec loci. And you find in bats it's expanded. It's an antiviral expansion in bats. What you'll find is that there's selection. Certain genes that cause a highly aggressive inflammatory response are missing, but there's selection in other genes. You can see loss of function. So you can see selection, loss, and gain of innate immune genes and antiviral genes. So there is a signature within the bat genomes. Because our genomes were so well assembled, what we also found was evidence of these endogenized viruses. So historical evidence in bat genomes that they had experienced and survived different types of viruses. So bats do have tolerance to these. So genomes can give us this information. What are we going to do next? Well, right now we're sequencing the genome of 6 of the suspected reservoir hosts, closely related bat genomes right across the Rhine-Lopid clade. We're working on that. And again, we want to ask the question, can you see any unique signature of innate immune response that allows bats to be tolerant? What we have to do, tools we need, is we need to develop the functional cellular assays, the cell lines, and the single cell transcriptomics to do this. You can do this for humans. So why not do it for the bats to try and work out, we need these tools to understand how bats can tolerate and live with viruses. So when the next pandemic comes, we have a better ensuite of tools to help us. But also the most important thing is that you need to network. And you have to network together. And this is just an example of GBatNet. It's funded by the NSF, bringing all different types of bat researchers together in one platform to answer the question of what is unique about bats.
Emma, we have another couple of minutes. Sorry to interrupt, another couple of minutes, thank you.
Last slide. So the thoughts that I want to leave you guys with is bats, are they the cause, the cure, or the future? They're probably all of the above. And to achieve this, to do what I hope we can do, first of all, we have to integrate. Scientists have to integrate together, all different types of sciences coming together to answer a question. And we have to network. So we've got to build the tools that allow us integrate and network. And this can come from the likes of using Slack, for example. Now we can all use Zoom, but we have to integrate better. We have to collect samples throughout the entire world, and we have to coordinate the collections and the databases. We're going to have to innovate. We need to come up with new methods to do this, from the sequencing methods to the preservation methods to the analysis methods. We have to translate all of our findings and translate them in terms of potentially, you can translate what you find unique within the bat immune system to allow us make better ways of treating people who are sick. You got to translate this into some type of meaningful context for managers. But one of the most important other things is that we have to educate, and we have to educate everybody. And again, by pulling us all together in this framework, we can look at not just the bats, not just the coronaviruses, but we can use this type of information framework to hopefully predict, stop, and prevent the next pandemic. So I just want to say thank you all for listening.